Hey guys, welcome back to a new video by Biology with Zhangshan. So today I'm going to talk about the Cambridge IGCSE Biology Paper 4 Theory Extended. This is the October-November 2024 Paper 4 Variant 1. So likewise for any of my past year paper videos, there will be a lot of responses that you can see there. And just want to let you know that in my responses, they are mostly in point form, but during exams or any practice, please get familiar with yourself with writing all of it in paragraphs. And also, most of my answers here are just suggestive answers that I think that commonly we will always put. But remember, there are also answers that from your side can be also accepted too. So make sure to cross check with the mark scheme constantly, and before commenting, or feel free to just again put down your answers if you want me to check. I'll be happy to, and this PDF will be put inside the Google Drive, where I will link into the description box. Okay, so let's start off with the first question. So they say figure 1.1 is a photomicrograph of a cross-section of a part of a leaf. So we come back to another question from the plant nutrition. So let's see for A part 1. State the letter in figure 1.1 that identifies where gas exchange is controlled. So the answer is L. So why is it L? Because L is actually the stomata. Okay, so L is actually the stomata. So although they ask you to put the letter, but you must also know like what is it lah. Statistically in your brain, you must know what is it. Most photosynthesis occurs at M. Okay, so M is actually pointing to you the I think there's a little bit of a confusion here. This should be N, because most photosynthesis happens at the palisade mesophyll cell, and the spongy cells are usually found at M. Okay, so I just realized that I just made a mistake. Why? Because you can see that usually for palisade mesophyll cells, it's lined quite parallel with each other. You can see the parallel sequence, while the spongy mesophyll cells usually have a lot of spaces in between. You can see the spaces in between here. So that shows that there's larger surface area for gas exchange to occur. Okay, apologies for that. And part 2, draw a circle on figure 1.1 to identify a vascular bundle. Remember, a vascular bundle is quite circular in shape. So the blue one that I circled, that is the vascular bundle with the xylem and the phloem inside. So translocation occurs in the vascular bundle. State the name of the two substances that are only moved by translocation. Well, it's quite simple and straightforward. So sucrose and amino acid is sufficient enough to answer this question. For part B, explain how xylem vessels are adapted for their function. So remember that they are asking you for their adaptation, meaning their structural characteristics, not the function. Okay, we know that the xylem functions for transporting water and also mineral ions, but mainly they want to tell you what is that characteristics that this xylem has that can help with the function that I just mentioned just now. So first, you need to remember in your brain that xylem vessels are dead cells. They have dead walls. Everything around them is dead. So when they are dead, right, they are usually very good at supporting the plant. So that's why first they have thicker walls. They have lignin. Lignin is actually the main material that is found inside xylem, which is actually dead cells. And we have function as a support of the plant. This has to come in your mind every single time, that anything that is dead usually helps for structural support. Okay, anything that stabilizes the structure of the entire component. Okay, and one more extra point, that cells are joined from end to end in like a continuous tube. It never ends, and that shows that it constantly pulls the water up from the root hair cells to the main plant. Okay, part C. Describe how water moves through a plant from the soil to air spaces in a leaf. So this question has already given you the clue that it shows that water moves through a plant. So first thing in your mind, osmosis. And when they never tell you directly osmosis, please write that in your answer. So water enters the root hair cell by osmosis, and they move from the root cortex cells to the xylem vessels, Water is moved out of xylem in the single direction. Remember, they only can go up, they cannot go down again. And it's held together by forces of attraction like cohesion. Cohesion between water molecules, then they will diffuse into the mesophyll cell as the moment they reach to the top. Okay, so just very simple. Just use what you have understand from osmosis and apply from how they move from from the soil to the other parts of the plant. Explain why plants need nitrate ions. So, well, if you forget, just remember that nitrate ions has the chemical element N. And remember that in proteins have these elements: carbon, hydrogen, oxygen, and nitrogen. So same thing, protein and nitrate ions, they are closely related to each other. So if we use protein for growth, same thing for the plants, they need to use to produce amino acids as a precursor, and they help to make proteins, and they will use it for the growth of the plant. So same thing, just translate it from a plant to from a human to a plant. That applies the same concept. It's just different chemicals, chemical stuff that's been put inside. Okay. Next question. They say question 2A is a chlamydia. It's a sexual transmitted infection that caused by a bacterium. Figure 2.1 shows the number of chlamydia cases in the country in 2018. So well, they have given you this graph, so you just have to look briefly. So calculate the percentage decrease in the number of chlamydia cases between the age groups 20 to 24 and 25 to 29, and give your answers in two significant figures. Well, another key point they have to tell you, and again percentage decrease. So well, we see another percentage decrease. First thing that we have to go and look for is how is the difference? Did it drop? Did it increase? So you can see between 20 to 24 to 25 to 29, there's a drop in the value, drop in the cases. So when you expect there's a drop in the cases, your value has to be in negative. Okay, so your answer has to be negative something 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 something. That is what you need to know. Okay, now two significant figures, and we're going to solve it now. So they say from between these two age group, we're going to get 3,900. Has to be clear in the number. There is no 3,910. No, because you can see the spaces are allocated for every 100. So 3,900 minus 1,600 divided by 3,900 times 100 equals to 58.97. Adjust the value to 2SF will be 59%. And because I mentioned before that it drops from one age group to another, so it becomes negative. So the answer is negative 59%. If you never put the negative, there could be a chance that you might get minus 1 mark. So be very careful in this exam. Part 2. Describe how the spread of STIs such as chlamydia can be controlled. So what we learn in the syllabus is mostly HIV. HIV is also a form of a STI. So you use the same strategies, intervention strategies for HIV towards chlamydia. Is the same thing. So first thing, of course, you know it's a sexual transmitted infection, you prevent sexual contact. You use proper protective equipment like condoms. Do tests and screenings if you think that you may be infected or you're worried of STIs. No needle sharing, and provide education or STI awareness. Okay, so usually this term of providing education is not very helpful, not just for these kind of questions, but also in questions in the environmental perspective in chapter 21. So B, chlamydia can damage the reproductive system. So state the name of the part of the female reproductive system that produces estrogen. So ovaries, okay? I do not know how to explain. You have to know that it's coming from the ovaries. So describe the role of estrogen at puberty. So they say puberty, not the menstrual cycle, okay? If they say menstrual cycle, then that could be another set of answers. But now they ask for puberty. So puberty happens most in girls with a lot of high amounts of estrogen. So you talk about what are the things that you can see. First, generally secondary sexual characteristics. If you have mentioned this, you're going to get 1 mark. Okay, then you explain all the features that you can see in secondary sexual characteristics. 1, there will be enlargement of the female breast. Then you can see the girls usually have more widened hips, and then they also help to kickstart the menstrual cycle, because in order for the menstrual cycle to begin, they need to have estrogen. Okay. Part C, complete the sentences. Well, this is a very common question you see since 2023, and you must expect at least one of these kind of questions in future examinations. So bacteria can be killed by drugs called as antibiotics. Well, this is quite straightforward. Make sure your spelling has to be correct. If there's one missing in your spelling, automatically you're going to get it wrong. So development of the strains of bacteria that are resistant to these drugs is an example of a natural selection. Well, I'm sure you know why it's natural selection, because they actually compete with who's going to be the best of the best, okay? And not artificial selection, because artificial selection is usually humans picking out the best characteristics, and we do not want that in bacteria. But generally, natural selection is a very natural process that it undergoes. Resistant strains of bacteria have gene mutations that enable them to survive drug treatments. And gene mutation, this is going to be the definition, random changes in the sequence of bases in the DNA. So this is actually the complete definition, and result in the formation of new alleles. A new alleles is a different version of the gene, okay, in the bacteria. Mutation rates can be increasing by ionizing radiation. So ionizing radiation is quite straightforward. Anything that can be very ionizable, like X-ray, gamma rays, okay? And in animal and plants, another source of genetic variation is a type of nuclear division. They tell you it's a type of nuclear division called meiosis, and not meiosis, because they ask you for genetic variation, okay? In order to have variation, you must undergo meiosis, all right? Okay, now we're going to go to the third question. Okay, they're going to link with you with chapter 1 entirely. So figure 3.1 shows the structure of the pathogen that causes cholera. Okay, so you can see they're going to link your chapter 1 to cholera questions later on. Okay, state the name of the kingdom that includes the pathogen shown in figure 3.1. So prokaryotes. I think it's straightforward. And now this kind of question happens a lot of time, and how they tell you this is 5 marks, so meaning that every column, every row is 1 mark. So let's say if you get one of these wrongs, automatically going to get 0. There will be no half marks here. So first, they tell you this is the flagellum. Flagellum is B. So what's the role of the flagellum? To move, alright? Like the sperm, they have a flagellum too. So what they do? They help to move. They help to propel the organism, okay? And cell membrane. Cell membrane is actually G. They tell you it's G. So this is actually what controls what moves in and out of the cell. You can also say movement in and out of the cell. That is definitely correct. So next one is ribosomes, which is at D, okay? So these are the rather small ones, which are D, okay? And not C, by the way. We're going to see why. So they do protein synthesis, and these are ribosomes. And remember, bacteria, they also have cell wall. So their cell wall is found at A, okay? You can see it's actually in the outer layer that is covering itself, and they help to provide support, which is actually the same function as what the plant cell wall would do, okay? And last one is plasmids. So plasmid, remember, plasmids are always circular. They are small circular ring of chromosomes, okay? And they do for genetic modification, which we actually see very frequently in questions. And for this, it will be C. Okay. Part B, describe how a person becomes infected with cholera. So it's just the straightforward answer of what are the causes of it. Okay, then just list one of the causes. You don't really have to elaborate that much. A person drinking contaminated water is sufficient enough for this 1 mark. Now, this one is where it comes up frequently, okay? So people with cholera can become very dehydrated. Explain how cholera causes dehydration. So remember, very dehydrated is due to the diarrhea, because diarrhea actually uses a lot of water. They're pushing all the water with the feces together. That's why when someone having diarrhea, you can see they are having what we know as watery poo. So explain how cholera actually causes dehydration. First, we need to talk about how the virus, the bacteria actually enters. First, they produce a cholera toxin, okay? Remember, most bacteria, they produce a toxin which can cause these sorts of symptoms that we can see. So they cause chloride ion secretion into the small intestine. Then there will be a loss of ions because you want to change the concentration gradient. When you change the concentration gradient, then you only allow water to move out of the cell by osmosis, of course, because it's water. Then this will cause diarrhea, okay? Because let's say this is your cells, okay, and this is your whatever things, the contents like that. What happens that because your ions are keep on losing, okay, they are going out, right? So what happens is that you are making inside here more high water potential. And when you make this side more higher water potential and inside becoming more solutes with a lesser water potential, what happens is that they will rush in here, and this will result in the loss of a lot of water. And D, vaccination can help prevent the spread of diseases such as cholera. Now you have to outline the process of how vaccination and explain how it can prevent the spread of disease. First, you talk about what is vaccination. Vaccination is a type of what? What kind of immunity they are producing? It's a form of an artificial active immunity. And what is inside the vaccine? They have weakened pathogens. They are not alive pathogens, they are weakened pathogens. So they won't cause severe symptoms in your body. So what happens is that these pathogens, they contain the antigens. They are found on the surface, and this can trigger an immune response. So these antibodies will bind complementary to the antigen, because antibodies are proteins. They have specificity, and they help to form memory cells, which actually helps to protect you from another reinfection of this pathogen, let's say. Okay, the question number 4. Complete the sentence about the mammalian nervous system. So we have the central nervous system consists of the brain and the spinal cord, and the peripheral nervous system consists of the nerve outside the brain and the spinal cord. Straightforward, alright? And now they've shown you the parts of the reflex arc. So we have to put the parts in the correct sequence, and this one, unfortunately, you have to memorize this. So first, it has to be a stimulus. Then there will be a receptor cell, and there will be a sensory neuron. The sensory neuron will send to the relay neuron, and they will send to the motor neuron, and then to the effectors, and then lastly, your response. So I think a lot of people may realize that why is it P? Because remember, when you have a stimulus, all of your tissues have receptors, and these stimulus, they will start to bind to this receptor cell, and this will only help to trigger certain responses that we can see. And next, state 2 stimuli that are detected by the skin. So we have temperature, touch, okay, or pressure that works, okay? And part 3, the pupil reflex controls the amount of light that enters the eye. State the name of an effector in the pupil reflex. When they ask for an effector, they ask for the name of the muscle that conducts this pupil reflex. And well, it's obvious that it could be the circular or the radial muscles. They either help you to constrict the pupils or they help to dilate the pupils. This depends, okay? And C, describe and explain how impulses are only passed in one direction from one neuron and the next. So they just tell you that there is a neuron, okay? And then there's another neuron. And how is this neuron passing the impulse at one direction only? Okay, and this is how you explain. In the synapse, the presynaptic neuron will release neurotransmitters from its vesicles. They will move to the synaptic cleft by diffusion. Okay, and neurotransmitters at the synaptic cleft will bind complementary to the receptors at the postsynaptic neuron, and then impulse is stimulated. That's it. 5 marks. Very simple. Just mention this kind of stuff. You are safe. All right. Okay, now genetics. All right. So the gene for red-green color vision is on the X chromosome. There are 2 alleles for this gene. So remember, this is actually just a normal. All right. This is a normal red-green color vision. This tells you this is a X-linked recessive, okay, or sex-linked genetic disorders. So the allele for the normal color vision is represented by the letter big B, and red-green color blindness is represented by the small b, which they tell you is going to be a recessive, and this is the dominant. Remember, when it comes to red-green color vision, your mind has to tell you this is an X-linked recessive. Okay, so now they've given you this pedigree chart. You may have to analyze this properly and probably take some time to actually go and see how is this being caused here and there. And you can see that in an X-linked recessive, males have higher chance in getting the disease. Generally, in most X-linked recessive, male have a higher chance. Why is that? Okay, so now the first question comes to this part. Explain why all the male children of parent 1 and parent 2 are red-green color blindness. So because parent 2 is an homozygous recessive, and only parent 2 can provide the X chromosome to the male offspring. Okay, well, that's quite straightforward. So That's the reason why. Because let's say if this is affected, let's say this is a female, right? A female with colorblind. So meaning in here it should be XB, XB. So when why all the males from these parents getting it? Because the B is passing on to the male, because the male only just needs one of these. Okay, they just need one of these small b's, and they are already colorblind. That is straightforward. Okay, and only parent who can provide the X chromosome to the male offspring. Okay. So here, this is just the way how you create a genetic diagram, and they give you parent 3 and parent 4. So you may have to decipher why is that, which is actually quite straightforward. It's just that they have already given you the phenotype, and you just plot it, all right, and then show your working of how you conduct this Punnett square, and then as a result just fill in all the information and make sure you put your probability. They say the probability of having red green color blind, so you can just see based on this and this, and you know it's already 50%, or you can also put it as 0.5. If you have any question on this, please do type out in the comment section below. We'll very much help you in this. Okay. And question 6. Penicillin can be produced in fermenters. State the name of the type of organism that produce penicillin. So just put fungus. It's really correct already. Okay, they say the type of organism, not the name of the organism. So if you put penicillium, it's wrong, because they ask you for what is the type of organism. If you know that penicillin is from a fungus, just put fungus. Don't have to put the name penicillium, because they ask for the type of organism. Alright, so the next graph is quite familiar. They ask you for the change in biomass and when grown in fermenter, and they show you this is actually an exponential graph. Okay, not an exponential graph, but a sigmoid curve. So this is actually known as a sigmoid curve. And how would you solve this? Okay, so they ask the name of the growth stage. Okay, so X is actually the lag phase, so a Y will be the log phase, so you can put exponential phase, is also okay, and a stationary phase. So make sure your term stationary is actually A and not E, because if it's E, it's all your pencils, your pens. Okay, so again, spelling error is important here. If you have one spelling error, then you're automatically going to get wrong for this part. 3 marks, you do not want to lose that. Figure 6.2 is a diagram of a fermenter containing the organism that produce penicillin. And you can see that the first question, I think it's a very familiar question you can see in practice. So explain why air is supplied to the fermenter shown in figure 6.2. Because air, they contain oxygen, they contain nitrogen. So what's all this for? It's for the organism requiring for oxygen. And what do they do with the oxygen? Like what we need to do for the oxygen? Yes, we do for breathing. But for the muscles, is to undergo aerobic respiration. So again, this is another question that you have to be careful with your words. You cannot just put respiration. It has to be aerobic, because aerobic means that it needs oxygen. It needs oxygen. If you just put respiration, I can tell you it can be anaerobic respiration, and anaerobic respiration doesn't use oxygen at all. So you have to be very careful with your term. If anything that relates with oxygen, just put that aerobic respiration. That is always safer. All right. And explain why the temperature in the fermenter must be controlled. Because first, respiration, you have to undergo respiration, and remember that respiration is also somewhat an enzyme controlled process. Okay, so there's a lot of enzymes here, okay, and to produce heat, and we must make sure that the fermenter is always at optimum temperature of the penicillium to grow. If it's not at optimum temperature, it won't grow well. Same as our body, if we put at 40 degrees, we will die because our enzymes cannot function properly. So that's the same concept that applies here again. And part 3, state the role of the paddles in the fermenter. So paddles are found here, and you can see these paddles are actually, with the direction they show you, it's actually going round and round. So what's the point of having this? So first is to ensure constant mixing. When you are doing your juice, you see that a blender is actually spinning. Why is it spinning? Because you want to make sure that everything is mixed well, everything is evenly distributed. So there's always a constant mixing, and you want to ensure there's an even distribution of the nutrients. The temperature in the entire fermenter is constant. Okay, then what are the other two, other than penicillin, what are the two commercial products that can be made using fermenters? So well, these are the two common ones that you found in the syllabus. One of it is insulin for diabetes, and also for mycoprotein. Mycoprotein is another form of food that we eat. is coming also from mushrooms. Okay, so you can see they are under the same species. So don't have to complicate yourself. I understand that it may be very hard to think during exam if you forget what are these things, but I think if there's any questions in the near future, it should be insulin and mycoprotein, and that's well sufficient to already answer this question. Okay, so yeah, that's pretty much for this paper. So if you have any questions, please do make sure that you comment in the comment section below. If you need any help, I will be there to support. All right, so thank you so much for watching. See you in the next video. Bye bye.